Okay, hi everyone. My name is Yaoting, and I'm the host of this workshop. And first, I would like to thank you for your interest to my proposed topic. And yeah, so thank you first. And so may I ask you if you have difficulty to hear my voice? If there's any problem, please let me know. And yeah. So and at the moment, I noticed I have just about 13 attendees present in this workshop. So maybe later there will be more people coming into this room. So in the first beginning, probably if possible, I would like to first have to have first to know maybe your name and a brief introduction of yourself, and if possible, maybe a quick introduction of your own project, if you are involved with any C-total project, or if not, maybe in other animal, and that's fine. So, and yeah, so if anyone, actually, I'm not quite familiar with this Zoom management. So maybe, I mean, now I would like to know if anyone would like to introduce yourself first. Then please raise your hand, and I will turn on. I will give you right to broadcast to all the attendees. So yeah, okay. I saw. Let me see. Paul, are you there? Let me see. Okay.
Hi, Yaoting, can you hear me?
Yes.
Yeah, this is Lauren, standing in for Paul. Did you need a hand managing the hands ups?
Yes, yes. So could you let me know how, if anyone raise their hands, how do I give their right to speak in front of the attendees?
So you should be able to hover over their name and press allow to talk, and that will let them speak to the rest of the group. Okay, okay. So you are the one who will control who can speak, who cannot speak, right? Okay. You can. Okay, I can do that for you. Sure.
Okay, okay, that's good. Yeah. So yeah. So please don't shy. Actually, I just, as I mentioned in my earlier email messages, that I have a very simple idea and just want to introduce you my previous study result in the Caribbean, and I reckon probably my developed result might be helpful for some of you. So that's why I make this proposal to the symposium organizer. So yeah, so may I first see if anyone want to say anything about yourself, so everyone can know a little bit of our group. Yeah.
Yaoting, if you want to pick people to speak, I can allow them to talk and then switch it off as we move through.
Yeah, please.
So we've got Cynthia up here at the moment.
Okay. Yeah. Hi, Cynthia.
Can you hear me?
Yeah, I can hear you.
Okay, hi, my name is Cynthia Legue. I'll get things started. So I'm based in the US, but I work in the Caribbean coast of Nicaragua. And my interest in this workshop is we have some post-nesting hawksbill turtles that we satellite tracked. And so I'm interested in looking at their movements in relation to the ocean currents. So, and I don't know how to do that. So
Okay.
that was my interest.
Yeah, excellent. If you reckon I can be a useful person, please let me know. Yeah, I'm certainly happy to if I have the capacity, I can help you to go through all the technical things. There's no problem. Yeah.
Okay, thank you. I appreciate
yeah,
that.
no problem. Yeah, you're most welcome. Yeah, yeah. Thank you.
Perfect. So, sorry.
So are there anyone else want to speak?
Yeah, we've got 6 hands up, so we can work through them quickly if you'd like before you get into the workshop.
Yeah, yeah.
Perfect.
Great.
I'll bring them up in order that I've got on my chat.
Okay, great.
Sumedha, can you hear us?
Yeah, I can hear you. You can
Brilliant.
hear me?
Yeah, we can hear you perfectly.
Oh, fine. Thank you. Hi, I'm Sumedha Korgaonkar. I'm doing my PhD from Wildlife Institute of India, Dehradun, and I'm working on the nesting behavior and migration of olive ridley sea turtles on the west coast of India. So the solitary population basically of olive ridleys. And my interest in this workshop is basically I have what the data I have seen for the past 2 decades. So there is a major shift in the nesting season. So what has caused, okay, we know very well about temperature, increase in the temperature and all those things. So that has resulted into the major shift. But what is the role of this water currents is always was the question in my mind. And
Yeah, sure.
not being the, not knowing about all this water currents and how the movement is. And I was actually searching for someone to get into this. And with your workshop, I could understand it. Also, I am working on migration of this sea turtles based on stable isotopes. So that is a very first kind of a work that has been done in Indian Ocean best. So that is Arabian Sea. So these water currents, then I could probably link water currents, their migratory movement. Luckily, this year we have done even the satellite tracking in our course. So that all satellite tracking data, stable isotopes, and probably we could answer why there is a shift in the nesting season, that too in the summers. So initially they used to come in the winters. Now we can see they are coming in the summers. So what driving them to come to the summer season? So that is, I feel this workshop would definitely help me into that. I've gone a little bit about this one, about the workshops.
Yeah. Thank you. Yeah. I wish I could have the power to solve all your problems. But as you mentioned, what you encountered in India probably has been also encountered when I was in the Caribbean. No problem. So not just as you mentioned, maybe current is one of the major factors affecting sea turtles migration patterns, but also there are also other factors like temperature, salinity, or their prey, whatever factor. So certainly. So maybe after this workshop, if you can give me at least a quick introduction of your project, then I'm certainly to see if I can provide any help to your your project problem. Yeah, thank you very
Definitely,
much.
I would literally love to do that. So I will send
Yeah,
you an
sure.
email, detailed email about it. Okay, thank you very much.
Yeah, thank you very much. Thank you. Okay.
All right, yeah, I think next we've got Sean Williamson.
Yeah.
Hi, Yaoting. Thanks for the workshop. I'm really excited for today. A lot to learn. I'm a postdoctoral fellow at Florida Atlantic University, but as you can probably hear, I'm actually Australian. And I'm also a project scientist at Upwell Turtles, a Californian-based NGO. I currently work on a few different tracking projects, mostly with early stage lost years turtles in the Atlantic. We've been putting tiny transmitters on leatherbacks in Florida and loggerheads in the Azores and green turtles in the Cayman Islands. And we're really interested in understanding how currents influence these early small, early life history small turtles, because the data, especially from the leatherbacks, because whether or not they're staying within those surface current zones, or the small leatherbacks who are only 3 months old, they seem to be able to dive to quite deep depths, like over 40 feet, even at 3 months of age. So it's trying to figure out how the stratification of currents influences their dispersal, and is something we're working on at the moment. So I'm interested to learn from you today, and looking forward to the workshop. Thank you.
Yeah, certainly. Probably as you already know that some of the smaller turtle in the early stage, because of their size, their weight, certainly it depends on the environment, particularly the currents are quite great. So maybe you have heard of small seagrass prey or stay on the sargassum and the macroalgae, and the sargassum certainly follow the ocean currents. So certainly it is a great, a big factor that affect the small sea turtles' migration behavior, and so certainly if so thank you very much.
Perfect. Yaoting, did you want to get started with the workshop, and we can have people introduce themselves in the chat?
Yes, I can start right now, but I don't know. Let me see. Okay, we already have about 24 attendees, and so do you want me to start the workshop right now?
It's up to you. There are some other people with their hands up, but yeah, your call if you want to start now. If you want to give people a little bit more time, just let me know what you'd like to do.
Yes, maybe I would like to hear some more idea from our other attendees. So please let the next one to speak a little bit of their projects. Thank you.
No problem. Next up, we've got Jenny.
Hi, Jenny. Hi, Jenny. Do you hear me?
Yeah, can you hear me now?
Yeah,
okay.
Very good.
Okay, so I run Cancer Turtle Rehabilitation Centre in Australia, but I also do a lot of satellite tracking. And the last 3 turtles that I've actually tracked have been just under 3 years old. So I'm really interested in currents, because one of them actually travelled 3,500 kilometres. So I'm really interested in currents and things like that with these young ones that are going out there with satellite trackers on them.
So may I ask you, do you have any PPT attached on these young turtles?
Do I have, pardon?
Do you have any? Have you put anything on the animals?
Yes, we put it. Yeah. So we had the wildlife computers on them.
Okay.
And so we had the good ones on it. So we got the depth and we got the distance and we got all of that from it, which was great. And it went up on, we've got a platform here called Reef Tracks, and it went up on that. And one of them actually, as I said, did over 3,500 kilometres. But the really interesting thing also was that she was 3/4 of the way to either Vanuatu or Solomon Islands, and she did a big turnaround And that's when Tonga actually, the volcano blew. And then she was back in towards Cairns. And then on the 5th of February, she did a sharp turn back away. And that's when we had an earthquake here. So really interesting data on, you know, what's happening. But I want to know about the currents and, you know, what she was traveling on. We also had an Olive Ridley, the first one with a tracker rehab one up here with a tracker. And she actually followed the whole continental shelf from Cape York over to the Northern Territory. So super interested in your talk.
Thank you very much. Yeah, so that's why I thought maybe my topic will be have your interest. So thank you very much. Certainly I will do my best to let you know how can we find any physical parameter to describe the effect of currents to sea turtles migration. And certainly I'm happy, look forward to see if you can share your observation and after this workshop. Thank you very much, Jenny.
Thank you.
All right, next we've got Veronica.
Hi, Veronica. Hi.
Hi. Well, my name is Veronica. I'm from Costa Rica and I'm doing my master's thesis currently. I'm actually based on the Pacific coast of the country, and I'm currently studying hawksbill turtles, mostly in their juvenile stages. But I'm not using actually satellite data. I'm actually using acoustic telemetry.
Excellent.
To study their
Excellent. Yeah.
The area that we are studying is pretty interesting because it's affected by several currents like North Pacific current and South Pacific undercurrent, and all that tropical currents kind of collide here. So I'm really interested in seeing how that might be affecting the movement of my turtles.
Okay.
I'm also tagging in the Pacific green turtles, so turtles that are crossing the Pacific to come to this side to feed, and we're not quite sure why they do that, but we're tagging them and we're seeing that they stay in our area for long periods of time. And I'm also actually studying the effect that a coastal upwelling might be having on their movements too. So I know it's not quite like an ocean current, but it's also a quite interesting oceanographic event that might be affecting these turtles. And I kind of want to see how all of these can come together. And see what's happening here.
Yeah, great. Your problem is probably also a problem to most of ourselves. But so you have your acoustic sensor attached on the turtle, do you?
Sorry.
Do you have you said that you have acoustic recorders, right? So
we're using acoustic telemetry. So telemetry. It's monitoring a smaller area, but we have like a good array of receivers. So we have a large Gulf kind of monitor right now.
Okay, great. Actually, when I did my PhD in Australia at the Curtin University, actually my major was marine acoustic. So if I have the privilege, I would like to know more about your acoustic recording from your monitoring of these sea turtles, and to see if I can find any additional information from the acoustic recording. Yeah, thank you very much. And I will do my best in my presentation. So thank you, Veronica. Yeah, thank you.
Great, thanks for that, Veronica. Yaoting, we've got one more person who'd like to introduce themselves. Max, I'll put him through now.
Hi, pleasure being here today. I'm also working with Upwell as Sean is. I'm an undergraduate at Princeton, studying EB, Ecology and Evolutionary Biology. And my research at the moment focuses on young sea turtles as well, and mostly looking at their dispersal from Atlantic beaches in Costa Rica. But I'm really excited to be here today.
Yeah, thank you very much. Yeah, Max.
Great, that's everyone who's put their hand up to speak. Did you want to get into the workshop?
Yeah. Okay. So, so let me open my PowerPoint first, and then let me try. See, oh, I need to reorganize my screen. Okay. So maybe I need to, okay. So now I need to share my screen. Let me see, this one. Okay. Okay. And then, oh. Sorry, this is my daughter's iMac. Let's see. Oh no, not the one. No, not the one. Maybe the basic one. I have a good assistant, my daughter. Sorry.
This is Zoom, right?
No, not. Yes, please.
Okay.
没关系，你就直接弄 ，Yeah。
也立即结束了。
Yeah, just press just in case the recording will be interrupted. So sorry, let me try the next step. Yeah, please click. Oh no, not the meeting. No, this is not right.
Yaoting, are you able to share your screen from the button at the bottom?
Sorry.
Are you able to share your screen using the button at the bottom of the Zoom call?
The system says that the Zoom has to be closed and open again.
Okay.
To share this. So maybe I should shut down for a moment and my dad would come up again. Is that okay?
Yep, I can be ready to add you back into the chat. Is there anything people in the workshop can do to get prepared for the content in the meantime?
Sorry.
就是爱关掉，然后重新用。
Let me see. Oh, who can share? Okay, and then.
Okay.
Okay. Is it this one?
This one.
Oh no, this one. Yeah. Okay.
It's not working.
Okay.
Yeah, it's working now.
Can you see my PowerPoint now?
Yes, we can.
Okay, thank you very much. So I will start my presentation. So this is the first page showing the topic and some more detail about the workshop. So overall, as you can see here, mainly I will focus on this topic presentation, and later in the second session, I will try to answer your question from this presentation. And in the last session, if anyone wants to really see the data processing steps on Matlab, then I can work with you, but I need to move from this Apple system to another system, which is Windows system, where I have all my data processing on that Windows environment. So if I suggest maybe you can go through the first session, and if you have question or have any good idea that we can have the opportunity to cooperate in the future for any topics, then certainly you're most welcome to go through the second session. So this is my plan for this workshop. So overall, this topic involves two variables, which is the first one is the turtle's instantaneous displacement from one point to its next point. And certainly it takes time for not just turtle, but for us to move from one position to another position. So it takes time. And in physics, or in math, it is a vector. So likewise, when we want to know what's the impact of the effect of ocean surface current to our study target, such as sea turtles or dolphin or whales, the most important factor around the study target is the water body immediately around the sea turtles. And unfortunately, current is a factor, and not just only a vector. So not just only a vector, but also it's changed from one moment to the next moment. So it is a dynamic vector, difficult for us to manage or to really get a sense of how the currents affect the animals in any time. And like most of you, we want to know what's the correlation or the effect of these currents to the animals' migration behavior. And since these two variables or these two vectors are dependent on position and also dependent on time, so the end result of these two vectors combined and become the so-called correlation is also dynamic as well. And fortunately, it is scalar. It might define the variables. Yeah, so this is the quick introduction of the math part of these two simple, but in the data process, quite difficult, challenging. Yeah. So in the following, I will give you a general view of what is the problem, what we are looking at from a bird's eye view perspectives. So this is the inverse problem. In other words, we observe the sea turtles, and then we have the data, but we want to know what could be the causes affect or have an ending result. And certainly we have many causes affect the sea turtles migration. And like ocean currents or salinity or cyclones or plastic litters or whatever oil and gas exploration activity or the surface traffic on the oceans. All these could be factors affect our result. Yeah, so there will be another topic on how we can sort from so many factors and find the major factor affect the sea turtles migration behavior. And one of the technique is called the principal component analysis, in short, PCA. So what's the physics of involved in this topic? And as you know, our sea turtles, certainly it can move and actually move. But if you compare sea turtles with other marine mammals like whales or dolphins, certainly sea turtles are slow in swimming. So, but overall, it is an active organism. But sometimes, not just only sea turtles, like whales or dolphins, probably you have heard some whale species, when they were in sleep, they just flow along with the currents. And so when in that state, I define it as passive swimmers. So in other words, in the season or in the whole day time period, some animal might be active, but in some moment they might be in sleep, and maybe they will choose some particular water domain in order to sleep. And when? So it is really a big question. And what else is we want to know? Can we determine when they are active swimmer or when they are passive swimmers? And what's the criteria to distinguish these two states? So come back to our topic. Certainly, ocean surface current is an active and dynamic driver. A dynamic factor affect sea turtles' movement. And finally, we want to know their relationship or correlation between sea turtles' orientation and the water around the sea turtles. So this is a quick overview of the general introduction from physics viewpoints. So, as you know, there are many drivers, many factors affect sea turtles' behavior, like the environmental drivers or man-made drivers. I know some of you might not necessarily directly involved with the sea turtles migration. Maybe more interested how our plastic affect the animals migration, but unfortunately there are many factors that we still don't know how to quantify their effects to the animals. But overall, I classify the driver into two big categories. So when I was in the Caribbean, there was a hurricane Mayhew just happened when some of the turtles released back to the seas. So like this one, I call this an event. And unfortunately, I still have no idea how to find any particular feature or physics from the hurricane its effect to the animals. So, but just to let you know, we don't know how the turtle feels when they encounter extreme weather conditions like this one. Yeah, so this is what I have just mentioned. And as you know, some institutes have used marine animals as a platform for their ocean observations. So the basic sensor that I have to put on the sea turtle is just a simple device without any additional sensors. But I know there are also other additional sensors like depth sensors. And in that regard, then the final result will be involved not just three-dimensional monitoring of the sea turtles on the surface, but also you will see due to the depth sensor, it gives you an additional dimension to look at how sea turtles dive in the water. So when that happened, then they will be more difficult and technically more challenging. So this is what I can see from here, but at the moment, I haven't get involved with any additional dimensional data. Yeah. Certainly, there are also other factors. I call it the invisible factor or driver, like one of our attendees mentioned. She has the underwater recorders, and as you know, the major energy that can propagate over long distance is acoustic, the sound. So no wonder all the marine animals use their calls for communication and for their survival as well. But unfortunately, after our development, we have created much more unneeded noise in water, and then maybe not just maybe, I mean, it's surely trouble to the marine animals. So we have airgun seismic exploration, which I have involved quite greatly in the past. In the sonar operation from the military, offshore wind farm, pipe piling in water, earthquake certainly is a factor. And any, no matter whether it is a natural attribute or man-made noise, all this noise could have an impact to our animals. So I call these invisible factors or drivers. But at this moment, I don't have the data, and I don't know how to maybe if I have the opportunity to involve with this data. Sorry, probably it's Siri. Okay. So I use a cartoon to show you what is active driver here. So ocean current is quite powerful, so it can move the turtle around. And for turtles, she feed on seagrass. So seagrass can be regarded as passive drivers. So this is two simple types of driver. But what about other objects like plastic litters or sargassum, macroalgae floating on the sea surface? I don't know what's the impact to their survivals. Maybe in the future, if you know anything about this, I'm more than happy to know. So just give you a quick break and think about what we have just mentioned. So what type of driver are dynamic? When I say dynamic, dynamic means the object or the driver always change its value or influence in different times, and also it change at a different location as well. So what type of driver are static, like a seagrass, right? Comparatively, it is still, yeah. So let me see. Okay, it's about, we have spent about 40 minutes, maybe. I will stop for a moment after an hour. So what are the key drivers nearby our animal? So these are two big categories. And the driver could be different for animals in the open and the coastal waters. So how to determine the main drivers? I have used the PCA, principal component analysis, but this is not within our topic. If you are interested to this topic, I can show you how to use this technique. And so once we have determined the principal components, like for example, the current is one of the major factor, and say temperature is the second one, and we will then end up from this PCA to use these two factors to see how these two factors affect the sea turtles' migration behavior. But again, these two factors or principal components might be also dynamic, and so they can be different in different times and different locations for the same animals. So a simple question is what we need to put on the map. So basically, certainly if it is related to sea turtle migration behavior, certainly you like to put some geographic feature like coastline contour or other object. And we for the dynamic object, we have at least the ocean surface current or ocean surface temperature or ocean surface salinity, whatever data you have. But at this moment, I only look at the currents. And so our static focus is these two dynamic factors, the displacement, the instantaneous displacement precisely, and the concurrent ocean currents, which these two vectors are dynamic, both dynamic, and in general mathematical form, it is in this way. So just for your reference, how we express these two vectors in the mathematical forms. So so sea turtle displacement over time period. So this is a quick explanation, description of sea turtle. Say it moved from position A to position B. Certainly it takes time. So the speed will be then the displacement over time. Okay, so quite simple, right? But we need to ask ourselves, how long in time is enough for logically referring the displacement for our study target when comparing to with the ocean surface current? Is it seconds enough or hours, or we need to have a more longer time, like days or months? So this is when really put your hands on the data, you need to think about this problem. So to the satellite system Argos, in its website, you can see there are a few limitations you need to consider. For example, it gives you the best spatial resolution up to about 150 meters. In other words, if your recorded telemetry data is worse than this number, it's normal. And but it could a problem to your study. So and due to the weather, particularly in the tropical area, due to the crowd, when satellite just pass over the crowd, and the sensor you put on the animal could not smoothly or successfully transmit its signal back to the satellite, then you will encounter some abnormal recordings. So at least in order for a good result, the satellite need to have at least 4 messages in one satellite pass over the animals, and the data quality is indicated in the message in the field of so-called location classes. So you can see more detail from their website, from Argo's website. So as I just mentioned, due to the weather, there's no fixed or determined time resolution. In my case, I had void data over a month, and the base resolution is 100 meters. Yeah, and if you encounter a great section of void data, then you will see when you put your plot your sea turtle track on your map, you will see some in statistics we call it the alias, and which is abnormal, but unfortunately it is normal in the real world due to the weather, the bad weather. So this is my real data. As you can see, the second, the turtle, yeah, and the fifth one, they simultaneously encounter a great section of void data, and over a month. So when you encounter such a data, how are you going to proceed in your data processing? There will be involved some technical problem. If you are interested in this problem, I can tell you in other opportunities. So this is the real sea turtle telemetry data, and the major one is the so-called last message date, and the second one is location date, and certainly you will see the longitude and latitude, which is the major two field that will allow us to get its actual location on Earth's surface. So yeah, this is basically what you will see from real data, telemetry data. It is from the Argus satellite telemetry data. So there are two date fields in each register telemetry message, each row, and which one should be used for referring each location's time. So let me go back to the previous slides. As you can see here, the first one is the so-called what I call the last message. And if you still remember in my previous slide, it said that the Argo system says that in order to get a position data for the longitude and latitude, at least each pass, each satellite pass need to have 4 messages received in order to identify the C total's position. So the first one, the so-called message date, or in my turn, last message, it is actually the time for the last message. The second field is called the location date. And this date is derived from, say, from the 4 message. So this derived location date, it should be used as the date or time for referring the moment that is registered in each location for each monitored position. Yeah, so this is my, just want to remind you, when you see there are 2 date fields in each message, and which one should be used for the location. So you should use the second one. So in a general form, we have a displacement, and the displacement is obtained by using the second position vector minus the first position vector. So quite simple, right? Yeah, okay. But you need to have your mind that it takes time. So it involves a time interval, delta t. So, but let me ask you a question. So suppose you already get the displacement, but should we appoint this displacement for position A or position B, right? So I will let you think about this. And in general, there's no positive, no sure answer. But in practice, we assign the displacement vector at the position A. But there's no harm if you can. If you know this situation, then when you processing your data, and if you can accommodate this change, then there's no problem. Yeah, but just remind you, in math, we often look at every detail. So as you have seen, the telemetry data recall the each monitored result in terms of longitude and latitude. So we need to convert this unit into our accustomed units. And yeah, so this is about the unit conversion. We want to convert this longitude and latitude into, say, kilometers or meters, so we can further process the data and display the result in our accustomed way. So, but again, may I ask you, is it reasonable to adopt the, say, millimeter for the displacement and the second for the time, and why? So if you remember that I just mentioned the best Argo satellite spatial resolution is 150 meters. So certainly if you want to use the unit for the displacement, then certainly millimeter seems not quite reasonable. certainly you can use millimeter, but it makes no sense because you will not see any changes from your satellite telemetry data because it's algo system limitation can only give you the best resolution about 150 meters. And as to the unit for time, will you see animals like sea turtle have any changes within a second or not? Probably not, right? So in other words, when we want to use a unit to for describing our animal, sometimes we need to consider the actual situation of our study target. So what is the, let me see, I still have 5 minutes, then we take a break. So what is displacement? Whenever we want to do studies in science, then we need to have a theory as a base to define our physical terms, like the displacement here. So in statistics, I redefine the displacement in my way. So this is my proposed a term for the displacement. I call it mean displacement. So what is mean displacement? The mean displacement in statistics is about how a random walker or the animal at the time t will move from position A to its next position B within a time duration of TW. In other words, say the animal have 10 steps within an hour, and every step it takes have different probabilities. I think that's quite practical. For example, when animals like sea turtles, in the first step, he saw a lady around him, then he might have a higher probability to go approach to the ladies, right? So in the equation, you can see at every different step, it has different probability that will affect its steps. So in mathematics, the actual form should be like this way. So this is my proposed variable for this displacement. I call it the mean displacement. So when you multiply the product, or sorry, you added up all this product for every steps it takes within these 10 steps, then you get an overall displacement for this animal to describe this animal's behavior over an hour. So conventionally, I have not seen a vector like what I have just shown you in this previous slide. So conventionally, the mean displacement has been introduced in 2008 by some researchers, and unfortunately, it is scalar without any directional information. And unfortunately, considering our ocean surface current, it is a vector. I mean, it has it involves duration, right? And our sea turtle movement also involve duration, right? So we do need to have a variable that can accommodate, that can consider this directional requirement. So that's why I propose this term as description for the displacement. I call it the mean displacement, MD. So this is a short introduction of the first term, the first vector, which is the displacement. And later, I think probably it's about time we can take a break. So may I propose we take maybe 10 minutes break, and then we come back here. So right on 2:10. Is it okay?
Hi, Yaoting, just jumping in quickly. If people just want to take a break from their computer, you guys can stay signed into the Zoom call just to save everyone having to log back in and get the password again. So we can get back into it at 10 past the hour.
Yeah, so could you convey your idea to all the audience for me?
Yeah, I think, was everyone able to hear that? Cool. So we've got a question from Kimberly Yaoting. She's asking what will happen after the break. So what's the next step in the workshop? But yeah, you guys are welcome to come back to your computer as it is at 10 past, and I'll let Yaoting answer that question.
Yeah, okay. Yeah. So I need to switch the sharing, right? Let me see. Okay.
Yeah, Tang, you can, if you don't need to close your presentation, you can probably keep your screen sharing.
Oh, okay.
Yeah. And then Kimberly was asking what was going to happen. What's the next part of the workshop after the break?
Yeah. The following workshop, the following slide will be talk about other terms. The second term, like ocean surface current, and the presentation of the result for their correlations between these two vectors. Yeah, basically it's about this. And then lastly, I will keep the attendees opportunity to say if they have any idea to the workshop and any future collaboration opportunity. This is what's going to happen in the following sessions. Yeah.
Okay, great. Thank you for that, Kimberly. Hopefully that answers your question. Yeah, if you guys are happy to take a break, we can come back and recommence at 10 past or in 10 minutes from now.
Yeah, thank you.
Yeah, Ting, you would just need to unmute your microphone. Yeah, Ting, can you hear me? Yeah, so you just need to click your microphone icon in the bottom left to unmute yourself. It'll be in the Zoom window.
Okay.
Oh, there we go. I can hear you now.
Okay, thank you very much.
Great.
Okay. Can we start right away? Proceed on to the next slide.
Yep, maybe just give it one more minute. I think people are still coming back.
Okay. Yeah.
Is anyone back at their screen? Are you guys ready to go? Can you just message in the chat if you're ready?
I'm ready, and if
Yep, it looks like most people are back.
okay, great. So the previous one is about vector versus scalar. So that's why I redefine the displacement as the so-called mean displacement in the way the way I just shown here on this slide. So it's a product of the displacement, real displacement, with the probability for the sea turtle to move within a steps. And then after we sum up all these steps, and then we get the defined mean displacement. So MD is defined in this way in Mars. So so how well, it looks like when we present it any way. Here I show you what it could be looks like if we present the mean displacement. In Matlab, I certainly already have a script to plot these figures. So one of the ways you can plot, you can displace the displacement over the entire 30 and 60 degrees. And the other way is you present on the second row, you can present its displacement over space, right? So the first row show you how many times the turtles oriented into any angles. And then the second display is show you how the animals make across the two-dimensional space over any distance. So there's a difference between the first row and the second row. And the last row, the third row, show you how the mean displacement or the animal oriented in any direction over the whole track. And so you can see over different times, sorry. So the last one, there's a trick in plotting the last one, but just to let you know, there are many different ways. But instead of plotting all the derived displacement, which is MD, for the entire section, for the entire migration track, I divided each track into 5 sections, starting from milestone 0 to the final destination, milestone 5. So here, what you see here, for each track, you have 5 milestone results. In other words, so why I would like to divide the entire track into smaller sections. There's a reason, because I want to know, because at this moment, I have not plotted this displacement on the geographic map, right? So if we want to see how the animal oriented in any duration, in any section, then I can use this figure to compare with each track on the map, which will be also marked with the milestone. So for example, for the first station, for the first milestone, you see in the first row, most often the sea turtle only are oriented to the west, right? To the to your left hand side. And the second row show you how this animal step away from its original position over any distance away from its original position. So the second row give you a sense of how far within the first station the animals move from its original position, right? So in the following slides, I will show you how to use this information, this result in order for us to better understand their behavior in statistics. So this involves statistics. That's why in the following slides, I will talk about some statistics as well. So this is the definition of our first vector, mean displacement. So what about our ocean surface current? In the real case, for example, when I look at the file I downloaded from the OSCAR database, I chose the finest resolution, but unfortunately, the final resolution is still quite coarse when comparing to our marine animals. So I have already wrote the Matlab function to read the net CDF file, and I already put it on my Google Drive folders. So there are two major speed components in the ocean surface data. The first one is called the meridional speed component, which is north-south direction. And the second one is zonal speed component, which is west-east direction. So yeah. So what's the limitation of this data set? The finest time resolution is about 5 days, approximately. And the best spatial resolution is about 50 kilometers. So you can imagine the spatial resolution is quite not good when comparing to our sea turtles, right? So when we want to interpret our result, we need to really careful about this constraint, about this limitation of our available data. So, and this data can only describe for surface currents within a water depth of 50 meters. So basically, this is quite simple, right? But when you really put your hands on this data, you will encounter many difficult technical problems in order to really get what you want. So what's the relation between these two vectors? As I have just shown you quite quickly. Here, how did I come up with parameter that I want? I thought maybe these two extreme conditions might be a clue for you to understand why I used that physical term for describing the correlationship between animals' displacement and the ocean surface current. Let's see the first condition. Say the red arrow is the sea turtle's displacement, and the blue arrow is the ocean current vector immediately around the animals. So they have a very small inclusive angle. And in that regard, in this condition, I would expect that the animal must follow the current quite closely. And in the other hand, in different conditions, if the animal go away from or against the ocean surface current durations, like this one, like the second condition here, and it has a very great included angle, right? So I would say then probably because the animal go to a almost totally different direction. So I thought maybe this would be a good idea to refer that the animal have a low correlation with the ocean current. So when I look at these two conditions, then I thought, okay, maybe I can use a simple mathematical form to use as a term for describing the correlation between sea turtle orientation or displacement and the currents. So this is what I propose. I call it angular correlation coefficient, in short, ACC. And the ACC is actually the inner product in mathematics, the inner product of displacement here MD and ocean surface current OSC. And if these two vectors are unit vectors, then we can further express this inner product as the cosine of the included angle formed by these two vectors, which is the displacement and the currents. So this is what I have proposed, and let's see after I use this term how it will how you will see the end result. So why, as I had just mentioned, I derived the ND from random walk. It's a theory you will see in statistics for describing algorithm, whatever size it is. It could be micro object like a virus or cells in our body, or a big size whale in the ocean. So no matter what, when we want to look at the animal's behavior, I reckon you will somehow encounter, have the opportunity to see this random walk theory. So if possible, and in the future, if you have chance to involve with animals' behavior, I reckon probably you need to see more about what is random walk. So, and as I had just mentioned, both vectors have to be normalized with respect to their respective maximum value. There's a reason why. So why we need to normalize these two vectors? Okay. So what is the property of ACC, which is the inner product of two vectors, ND and the OSC? And the end result of this, in terms of the absolute values of these two vectors, its value varied from 0 to 1. However, its inner product, which is ACC here, its value vary from -1 to +1. So why we need to normalize these two vectors, ND and OSC? It's because each animal has its behavior, right? So when different animals want to be compared for their for understanding their orientation between two different animals, then we need to consider if we already have a standard scale. We need to have a common scale. You cannot say that you have a coarse scale to measure my height, and use a smaller final scale to measure your height, right? We need to have a common scale. So likewise, That's why we need to normalize all the number with respect to their maximum value. And after that, then we can use the normalized result to compare their numbers for different animals. That's why we need to normalize all the ND values for different for all the animals. So you may ask, there are many current vector on the surface, right? And actually, it just not only many vectors, but also it changes over time. So how do you find the right one, the individual one, the right one, right next to the animal? And unfortunately, the animal also move around from one moment to another moment, right? Because it is a living thing, it is a living organism. So you will wonder which vector to pick, which current vector to pick, and when to pick, right? And so it involves an algorithm which I propose for obtaining the right current vector in order to use the determined vector to calculate with the instantaneous, the concurrent total orientations, right? So it involves the identification of the right OHC vector flowing right next to our sea turtles, right? So, and certainly you will see it involves calculation of the mathematical form here, right? So, and yeah, so the end result, ACC is a scalar. And when I present the result for the ACC, unfortunately, it is also dynamic because our input are dynamic. So certainly the end result is also a dynamic parameters. In other words, it changes over time and also depends on where you get the result for the ACC, right? So that's why it's not easy to display this result in a still image. And so I generate a video for you to roughly understand how the ACC vary over different times and over different locations for different animals. So this is the finish of our result for the correlation description for the animals migration or orientation behavior. So I don't know whether I should show you the video or not, but it is, if you have not seen the video, maybe let me see. Oh, why? Let me see. Sorry. A moment. Maybe I need my. Sorry. A moment. The video. Let me show you the video. Let me see, where's my video? a moment. No, not this one.
Yaoting, while we get the video running, there's a couple of questions in the chat and Max has his hand up. If you'd mind taking his question.
Yeah, please go ahead, Max.
Yep, putting Max through.
Hi, Yaoting. I just had a quick question about a while ago you put the definition of mean displacement up on the screen, but I wasn't sure how you actually calculate these probabilities of going from xt to xt plus 1 in practice. Does that use the PCA analysis you were talking about before, or have we not got there yet?
No. As I yeah, so maybe my talk has some confusion to you. Actually, the ACC is just one of the parameters we can use for describing our animals' migration behavior affected by the currents, right? So it is just one of the many drivers, one of the many factors. So it has not yet involved with any calculation for determining which one is the key factors, right? So at this moment, I just show you one of the many factors, which is the OSC, the current at this moment. I have not tell you how to show you to sort out from many factors in order to get the key factor for describing our total behavior. No, not yet. Yes. So, but if you're interested to this principal component analysis, certainly I'm happy to show you how to do that because my thesis involve a great deal of PCC. Yeah. So, but anyway, please send me a message later after this workshop, and then maybe I can give you some clue about how to identify from many factors and then sort out to get to the major factor for describing our animals. Yeah.
I'm sorry. That's a good answer for one of my confusions. But I think I was also still confused about how you these probabilities that you use in your equation. How are you getting?
Yeah. Sorry, I forgot to tell you this problem. Yeah. So then I'm working on a manage from the result. So I forgot to tell you that unfortunately we don't have the power, or at least any way they can have understanding of how animal step from position A to position B. So in the actual calculation, I use a fixed probability for all steps, and it simplifies my calculation for me to get the final mean displacement of the MD variables. Is it answer your question?
So what probability distribution do you use? Do you is it just uniform, or does it
just uniform? Just uniform. Yeah, just uniform. Yeah.
I see. So yeah. In the end, then, is your mean displacement is just the total displacement then, because you can factor the probability out.
No, as long as I have chosen how many steps within that time duration, then I can know how many steps, right? So then I say within an hour there are 5 or 10 steps. Then I use 1/10 as the probability, as a common uniform probability for all steps within that time durations. Is it clear to you now? Or maybe maybe later, if you are interested to, could you send me a message later after this workshop? I'm still struggling to find out. Let me see the video. Let me see. I remember I put it on. Oh yes, here. Let me see here. Okay, so there's a V. No. No, it's not working. I don't know why. Yeah, okay, my video here.
Sorry, Yaoting. Max, maybe we can double back to that question at the end. Yaoting, there was one more question about the 5 sections you defined in your track. How did you split the track into the 5 parts just before we get to the video or the rest of the session?
Yes, you know, maybe, sorry, I was focusing on finding my video. Can you ask a question again?
Yeah, no problem. Jenna had a question about how you split the track example you put on your screen into the 5 sections with the 5 milestones.
Okay. Yeah, I just evenly divided every track section into individual sections. So there's a reason why why I explaining your question. Yeah. Okay. Can you show? Okay. Click on play. Do you see the video?
Yep, we can see the video.
Okay. So basically, I just evenly divided each track into 5 sections. There is a reason why I want to make this arrangement. That's because when we displace the track over different times, we cannot really understand how the statistic features looks like on the map, right? So on my map, I also mark these 5 milestones. So you can roughly, by comparing between 2 figures, one is the derived, say the ACC result over time domain, while the other graph is the whole track. But you don't know how the statistic result could be looks like from your observation of the track on the map, right? So that's why I would like to have the milestone marked simultaneously on the geographic map and also on the data display over different times. This allows me to see if there's any particular feature in any different divided sections. This is the reason why I put milestone for each track.
Okay.
Am I answer your question?
I believe so. Jenna can follow up in the chat if not, but just to sort of summarize, for the different tracks, you'd split them into 5 parts, regardless of how long they were, so you could compare across those 5 sections and individuals.
Exactly. Thank you, Lauren.
Right.
Yeah. Okay. Are there any questions from you for those slides I have presented? If not, okay, let's see the video. So, okay, sure. Please go ahead. Can Lauren allow Sure to say?
Yep, sure. You should have your microphone unmuted now.
Yeah, can you hear me?
Yes, please.
So when you calculate all of that, this is only one part of the route, right?
Yeah.
So like the turtle migrates like in a whole route, and this is like only with calculation for like small parts of it, right?
I think I not really get your idea. Sorry.
No, like okay, I'll enlarge that.
I think
like
no, you go ahead, Shay, sorry.
When you calculate all those small parts of the route, and you have like a bigger idea of how much the turtle is swimming against the current during the whole migration route, right?
Yeah.
Like,
yeah.
What percentage, like what are you looking for? What's the percentage of migration that is
affected by the currents,
sort
right?
of, yeah, affected by the currents or semi-planktonic or something like that? Like what's normal, what's not? Like how do you calculate that?
Yeah. I would not say normal or not, but to use the normal term, no. But I use the ACC here, as you can already seen from the video here. I use the color, the correlation, which is ACC here. I use for describing the correlation, and it is encoded in colors. So on the video, on the color bar, I use maybe orange or yellow color to indicate their strong correlations, and I use blue color for the moment maybe the turtle is swimming against the current. So from this from the use of this color indicated on the color bar, and see your monitor the sea turtles on the map on along the track. I have put all this color on the animals track, right? So the yellow color roughly tell you that the animal is following the current. It is flowing along the current, like you catch it. Say if there's a free shuttle for you to go to school, why do you give up the opportunity? It saves your energy, right? If you want to travel over distance, then you certainly want to have that free offer for you to move from point A to point B, right? So when you plug all this color, all the ACC parameter on the map, you can see how the animal was moving from its nesting site to their foraging ground in any kind of condition, which is measured by the parameters. Sorry, I will play it again, right? So the color, which is a measure for describing the correlation between the animal's orientation and the current, right? So I use color as an indicator for this measure. So when you see the color, when it is in yellow color, then you can somehow guess that maybe the animal just follow along with the currents. But when it is in blue colors, that means it probably only it is swimming against the current. But let me remind you in the video. So I just pause the video for a moment and say, okay, I have turtle 5.
No, I see the colors, and it's very clear. Like it's very, you can see it. It's very good.
Yeah.
But the question is like, what's a behavior that we're looking for? You know, like you see a turtle swimming with the current or against the current, but like, do we know like what's the natural behavior of turtle in certain areas, or like what do you want to compare?
Yeah, this is a good question, but unfortunately, this is what I also want to know as well. That's why in the following slide, I will raise my idea for your interest as well, and maybe you can answer the question for me. But let me continue what I want to tell you. Okay, on your right bottom corner, you can see the T file, right? So this T file, C total, starts from a small island called Tobago. And it started from this small island. When it immediately leave its nesting site, it immediately follow the current. And then up until somewhere, St. Lucia, maybe somewhere to its destination. Within the small foraging ground, you can see its ACC always randomly change from blue color to yellow color and from yellow color to blue color. So why is that? That means the turtle probably is located in a very shallow area, and the current probably do not have the power to really affect its orientation. In another possible condition is she find a perfect habitat, and it's just looking around and feed on the seagrass and in a relaxed condition. So when you just look at the T5, when she is randomly showing different colors from yellow to blue, from blue to colors, then we can see from this situation, and the guest probably she was happily enjoy her dinner. So, but we don't know whether it is true or not. But later I will show you in statistics how the orientations could be quantified for showing whether an animal is in a relaxed condition or not. Okay. So can I just maybe I stop here and then proceed on to my next slide. So I need to switch back to, yeah, okay. So, and actually I need to maybe quicker. Okay. So there are many technical issues you need to consider in order to really get a good result. So these are just some of the points you need to consider, like if you have a great section of void data, and how can you manage that void data. So one of the step is you need to resample the data and assume that within the section, within that missed section, within that void data section, you need to assume, in my condition, I just assume that the animal go along with a straight line, right? Instead of following a curved line within the section. So this also involves some assumption as well, but just let you know what I did. And certainly you need to have a logical unit for as a measure, as a unit for referring your measurement. So it also involves unit conversion, conversion and the normalization. So the pre-processing steps also involve smoothing. So in the next slide, I will show why smoothing property is necessary, and the resampling. And when resampling, we need to interpolate the data if you have large section of void data, and certainly normalization as well. So, and also the time duration for obtaining the displacement. But certainly, if you have a very good data quality, then this is not necessary. So I show you some of my study results. So for example, this is the distance profile that these 5 sea turtles, these 5 postulating host peer has completed within their monitored time period. As you can see, if the slope is quite steep, then that means that animals move very quickly. But why it moves so quickly, we need to know, right? So that's why sometimes, and actually in critical situation, we need to compare from one figure to another figure in order to figure out why it moves so quickly, and why compared to other animal, why it moves comparably slower, right? So, but at least this is just give you an overview of how the animal has traveled within their monitored time periods. So why we need smoothing like this one? As I have just mentioned, due to the bad weather condition, quite often the Argos satellite system cannot really identify the animals. So in the particular situation is the recorded data has an abnormal position quite far away from its track, right? For example, you can see on this slide, there are 2 points on your left-hand side, almost 100 kilometers away from its next step, which is quite impossible, right? In sea turtle certainly will not move so quickly away from its original position within just an hour or 2, right? So over 100 kilometers. So this is why I mentioned why you need to smooth your data. And in statistics, we call it the alias. And after smoothing, you get the red color track. And although it still have some difference against its original track, but still somehow follow the original track for the smooth track. So this is show you how why we need to interpolation. The left-hand side is the real data with approximation of void data, right? So this is the availability of the data over time, over their monitored time. So in order to fill up those void data session, I assume that the animals go on a straight line to its resume the track, which is the right figure after interpolation. And this is show you how the effect of the interpolation over time. And this is to show you how the effect of interpolation of their track on the geographic track, on the geographic map. And fortunately, the void data all happened within a very short range, a very narrow area, which I reckon probably within their foraging ground. And yeah, so you do not see if there are any different major difference between this before and the after interpolation on the geographic map. So this is just show you why we need to do the interpolation and the smoothing. Yeah, so let's go on to the next one. It's about resample. So why we need to do resample? It's all about the statistics, because say if say in an extreme condition, say you only sample, you record, you register the data for once an hour, and then the next moment you record the animal 10 times an hour, then that's not fair, right? So in order for have a same scale and have a same number presentation for all the animal to be displayed on the same scale for how many times they have visited the one location, you need to have a fixed sampling rate. So this is why we need to do resampling. So in this figure, since I want to know how often each animal stay within any locations, so I use the frequency, the visitor frequency. So after I have a fixed sampling rate, I can put all this visitor frequency for all the animal and to put on their individual maps. So this visitor frequency for how many times they stay within a small section on the locations was coded in color. So as you can see, I also use color bar by the fifth turtle 5, T5, right? So there's a color bar here showing how frequently the animal stayed within any specific locations. And it allowed me to know or roughly understand whether that particular hotspot is really a hotspot or not, right? So I use the interaction of two dashed lines. For example, for T5, I use the vertical line and the horizontal dashed line to let you know the interaction of these two dashed lines is actually the which has the highest visitor frequency. So I use this highest visitor frequency, this highest number, as an indicator to indicate whether it is the foraging hotspot or not, right? Is it readable or not? I reckon it is readable. This is why we need to have a fixed sampling rate. This is why we need to resample the data so that we can have a result like this one, and put all our derived result for all the animal and using the same scale, which is the color as shown in the color bar, right? So this is one of the important indicator we can get from statistics to identify where on any location there might be a key foraging ground for that animals. Okay. So another section here, another slide, sorry. So another reason why we need to have a normalization, we need to have a common scale for comparison between different animals. And I further divided each track into evenly divided section, which I use milestone. So starting from milestone, as you can see from the top of the first panels, that each milestone starting from milestone 0 to milestone 5 is destination. And I show all the results like the speed on the left axis. I show the statistic result of the speed statistic, and also the another parameter I derived from random walk, which is so-called straightness index. So I calculate its frequency, and I found some interesting result. I will say more about this, and we will have chance to come back to this slide again after I show you the following slide. So I propose another parameters. So I propose this model for describing animals' behavior in relaxed status. Why I want to have this model? Because as I have mentioned in my previous slides, I found that some animal stay quite often, spend quite a long time within quite narrow area. And I reckon they must be in a relaxed state. Why? Because from the ACC randomly change over time within the hotspot. So I reckon probably I can use some mathematical model to characterize whether it is in relaxation or not. So I propose maybe the so-called in math in physics, we have a so-called exponential decay function like this one. So for this quantitatively estimating the most likely speed v of animal in relaxation, I assume that the speed distribution of sea turtle when it is in feeding should follow the above in statistics. They termed in exponential probability density functions here, as I show you here. Okay, so maybe I can skip some of the technical or mathematical property. And then if you are interested to this model, I will tell you more maybe later, because I found I have spent another hour. So I need to quickly go through here. So what is my expectation? Let me see. Okay. Okay. So let's go back to this slide. You can see my vertical axis, one of the major is to show you how the visitor frequency within each section and the horizontal line indicate the frequency values. In other words, most often, say in the case of T2, the second row, you can see most of the time, based on T2 speed distributions, it stay in a very, it spend most of the time maintaining the very low speed. So from its speed statistics, you can see it seems follow a so-called, what I so-called exponential decay model. So I use the model to fit this observed result. And in that model, there is a so-called characterization index, which is new here. And so in T2, in the second row, you can see I divided its monitor track into 5 sections. So after first section, I found that it seemingly followed the model quite perfectly and have a very uniform characterization parameter, which is new here. So and when I look at its condition, its track on the map, I found that T2 was maintained a very randomly orientation condition within the hotspot. So that's why I want to divide the track into smaller sections, like this one here, 5 sections. And I found that this animal, this T2, have a very uniform mu values, which is starting from the second section 0.07, 0.09, 0.08, 0.09. Likewise, for T4, the fourth animal, it also seems has a very uniform pattern, except in the second section. And likewise, for T4, it seems that these 3 animals all have a similar relaxation behavior following the exponential decay models. So if eventually, if my observed species, host bear, if it can has a stable mu values, right? Then eventually, what I expect from this result is, if we can have the mu value for different species like whales, like dolphin, like seals or turtles, then if their mu value here, which is indicated on the vertical axis, although they might have overlaps between different species, and say in the future we can have a catalog that can derive from this assessment, then we can use this catalog as a reference for identifying different species indirectly. Actually, I'm a person like to know things indirectly instead of from your appearance, from the behavior, from the statistics science. So this is one way we can further investigate to see if we can, if we finally, fortunately, if we can have a catalog like this one and showing a map like this one, then we can use this map, this chart, as a reference for the species identification purpose. So there are still a lot of possibility we can go from here. So the following section, I will quickly go through, and then I think I should conclude my workshop. But just to let you know, there are also other opportunity, different way we can present our result. So here I show you how to show movement on a still image like this one on a geographic map. Like this one, I use milestone. So as I have mentioned, if I compare this map showing different milestone and comparing against this one and against let me see if and against this distance profile, I can roughly figure out how each individual animal will move in from any position to its next position. in any way, right? So I compare this one and with this one, right? All marked with milestone. So as I had mentioned, T2 spent most of her time within a quite narrow area, right? And for likewise, T4, she spent quite a long time of Guyana. And unfortunately, there's oil field just recently found recently. And I will tell you more about the possible impact of this oil exploration so close to the hotspot. And in T4, you can see this animal also spent quite a long time indicated by all the rest of the milestone within such a small area, right? So that's why I say there's an advantage into check. Then we can compare steering with
Yaoting, it seems like there's a bit of a lag with your internet.
Oh, yes. I have cable. I don't to show you this of who are only this trick, because actually the horizontal domain is time, and the vertical domain is the north south direction orientation scale. So actually, I did not show you the horizontal displacement scale. So when you want to show your displacement this way, you need to rearrange the scale in comparison with the time scale, which I presented here. So how to show the variation for all variable over time, right? So again, I divided all the section into 5 sections. The first row is a straightening index I derived from random walk. The second row is about the speed. It's a speed variation over time. So again, this is also a good example of why I use milestone. You can see the second row from the second row, right? Its speed vary, right? So when you compare this speed profile and compare this speed profile against say these figures, then you can roughly estimate how fast or how slow the animal was moving within the first section. Sorry, yeah. And the second, the third row is about the angular frequency, for how frequently the animal oriented to any direction within each section. Likewise, the fourth row is about how far in spatial domain within each section the animal move across any spatial domain, right? So there's a difference between row third and row fourth, right? So it all depends on what you want to present. But just let you know, there are many different ways you can present a result in whether in time domain, in angle, or in spatial domain. And the fifth role is about the displacement variation over time. It's nothing. And the 1, 2, 3, 4, 5, 6. The sixth role is about the ocean surface current, right? So you have the fifth role, which is the displacement, and it's follow the role, which is the environment, the current, and you can see their orientation, right? Roughly, and then you see the result, which is the bottom row. You operate this mean displacement in the fifth row with its environment, which is current, OSC, and you get the bottom result, ACC, the bottom row. Whenever the ACC is high, has a high value, then it indicates to you that the animal will follow in the current very closely. And this is one of the advantage or value you can see from this result. So, and this is just show you how to show all the variables variation over time. So how to put all these things together on time domain? Okay, that is one. If you just want to show these three variables, the displacement, the currents, and their correlation values, right? So the vertical axis show you the scale of its northing amplitude, and the vertical scale, which I use blue color, to show you the current amplitude. And I show you the result, which is ACC, their correlation on the top of each panel, right? So this is one of the ways you can put all these three key variables that I want to show you in this workshop. And yeah, and the ACC its value was coded in color, as you can see from the color bar in the bottom, on the bottom of this slide. So if you look at its orientation coded in black color, and its environment, which is currents, in blue color, and finally see how their correlation over time indicated by the color bar in the bottom. Yeah. So okay, I think I spent almost another quite long time. So I want to quickly go through the following slide. So there are many steps I have been go through, and so let me see. Probably this about, and so one of our attendees used to ask me how to generate a coastline contour for free. And yeah, so I already sent you the answer in my previous email message. If you have watched the emails. Okay, so finally, I want to use this slide to conclude my workshop. So from my study, I reckon there's a more big picture, we can say, for example, where the animal might meet the oceans, and when, and how the animal was meeting with the currents, and how we can measure their interaction, their impact, whether it is plastic liters or whatever driver factor that affect our organism, is quite an important and big issues. And before I wrap up this workshop, I would like to have your attention that I have just mentioned, that T4, my fourth animals, unfortunately, her destination is quite close to the big oil field, just off Guyana's capital, Georgetown. And I don't know whether it is fortunate or unfortunate, that because Guyana is expecting to be one or two of the major oil industry player. But at least I can see the nearby habitat surely would be subject to the oil industry activities. And what I also have found from my study is that there's a telltale sign that, from my understanding of oceanography in that area, around the Trinidad and Tobago, most of the time, the currents only flow upward to the north. You will not see often that the current is flowing to the south. And but the T4, this female, when it left its nesting ground to the east side of Trinidad, it stayed in the coast water for at least a few weeks, but it did not follow the nearby current to the north. I don't know why, but there was a moment when the current changed direction down to the south, to the southern America. It catch the current and immediately swiftly flow along with the south moving current to the coast water of Guyana. It is quite interesting why T4 don't bother to follow the north flowing current. Instead, she just follow, he catch the short period of time, a very short window when the south moving current moved by her. She catch the opportunity and immediately swim from the coast water of Trinidad down to area close to Guyana. So from that indication, from my observation of T4, I suspect that maybe it is there's a memory in T4. She will come from that area, so she would rather go down south instead of go up to north. The other possibility is probably it's due to our nature, because all the animals want to diversify their population over different areas, so they can increase their survival possibility. This is another possibility, but we never know. But from this observation from T4, I saw there are a lot of still have a lot of questions remain, and I look forward to see if we will have any good idea for how to get this question answered. So, and let me see if I haven't. Okay, I just finished my first session. Okay, so okay, I will go on to the next session, and then yeah. So I finished the first session, then did the and I want to know if any of our attendees would like to show your idea. So you have any question, you are most welcome to raise your question now. Thank you. Lauren?
Yep, I'm here. Thanks for that. That was really interesting.
Sorry, do you hear me? So yeah, so do you hear me? And can you help me to raise those people who want to speak?
Yeah, does anyone have any questions, or are we keen to move into session 2? If you're keen to move to session 2, just let me know in the chat as well, so we know where everyone's at.
And also, please help me to see if there's any question in your chat room, in our chat room. And if at this moment you don't have any question, certainly you're certainly welcome to raise your question in your future email communication to me. And if I have the capacity, and certainly I'm more than happy to let you know what I think. And certainly from as I have mentioned in my emails, that I regard this workshop as an opportunity for us to form a community interest group on this sea turtle monitoring project. So if you have any idea that you want to share to us for any future collaboration opportunity, please let me know, and I will send your message to other participants to see if there's any opportunity for all of us. Yeah, thank you.
Yeah, that sounds great. It doesn't seem like anyone has any questions at the moment. If we want to move to the next part. Oh wait, we've got one question coming through now.
Oh yes, sure.
Here we go.
Can you hear me?
Yes, yeah, very good.
Thank you. This was very good for just to start my my question with. This was very engaging and very good. And my question is, like, you showed one individual that you compared with the currents and did all the equations, but I guess that you have like number of individuals, and each of our researchers contain more. So is it like you made this way to do all of the analysis in a like algorithm or something that you can compare all the individuals in the research, or is it like
okay. I think my quick question is, let me show you the mathematical models again here. Why I propose this model for describing, actually this can be used for describing any organism, even bacteria or big size whales, no problem at all. This model can use for describing whether the animal is in a relaxation state or not, by looking at the so-called characterization parameter, which is mu here. Let me see here. Okay, here mu. The mu on this chart, you can see whenever the animal stay within a small area and have the highest frequency of slow speed. That means when slow speed means it does not move much within that narrow area, then it must be in a relaxation stage. why they move so slowly within that narrow area, right? So I want to use a model that we can follow as a base for all different animals. This is why I use a model. And then from the result I got here, the mu value here. Then, since I already found that this postulating host sphere all have almost uniform mu values. So then if you look at this chart, this is my expected result. I found the mean speed, which is mu here, for turtle. It has value with this range, right? This is for our post-nesting host sphere. Maybe different turtle species has different mu value distributions. But if in general, the turtle have a mu speed distribution range like this one here. And if other animals like seals, dolphin, whales, if they have their own distinctive mu value distribution range, then we can use this chart as a reference to indirectly identify if the monitored animal is any kind of animal, right? So this is what I expected. And this surely could be done if we had more data from different biological groups doing their own research for different study target. Then they will be they will provide an additional reference for us to identify indirectly whether the monitored animal are any kind of species, right? Say if you have your whale data and I have my turtle data, and if we mix all the data together, and if we can see two clusters of data shown on this chart, then if I have the power to enclose the data within the whale, then I say, okay, maybe this data is coming from where, and the other cluster maybe are coming from the turtles. So in that way, that is why I say there's a usefulness of this chart. If we can all have different data sets come together, maybe we can generate a chart like this one for biology to indirectly refer if the animal is a turtle or seals or dolphin or whales. Yeah, I hope I answer your question here.
Yes, thank you.
No worry.
Did anyone else have questions they wanted to ask?
If no more question, I think we have spent quite a long time. And as I mentioned, we all have limited energy and power. And I reckon some of you might be living in a different time zone, so must be quite tired. But overall, all in all, you're most welcome to let me know your question later after the conclusion of this workshop. If not, and if there are any person who are interested to the real data processing on Matlab, and then certainly I'm happy to show you those scripts, but they will involve a very great deal of time. And so if no more question, Lauren, I would like to thank you, and thank you for all the attendees have the interest. And I want to say again that I really I feel honored that I can present you my result here. although I have not published the result. And I also would like to let you know that I thank you for those my colleagues in the Caribbean at the University of Trinidad and Tobago when I was with the University of Trinidad and Tobago in 2015 and 2018. Yeah, so I love the Caribbean. I have to say so, but it was a mixed feeling. But thank you very much, Lauren, and all the attendees. Thank you very much.
Thanks, Yaoting.
So, yeah.
Did you want to run through the demonstrations in this Zoom call or separately?
I have no more slide, but I will put this PowerPoint file onto my personal Google Drive and share this PowerPoint to all the attendees or any other people who are interested to this topic for their suggestions. So yeah, I think it's about time I need to conclude this workshop. Thank you.
Great, no problem. So, thanks everyone for attending as well.
Yeah, so I want to close. I've stopped my sharing and leave this workshop. Thank you. Bye. Hi everyone. Bye.